I'm very excited about this next talk because it's going to talk about breakthroughs in biology and using microbiomes as agents. And we are thrilled to have a very special guest speaker here. He is a professor of microbiology or bioengineering at Stanford University. Put your hands together. Help me welcome Professor Michael Fischbach.
How are you guys? This is an unusual experience for me, but I'm going to try to tell you a scientific story without leaving any detail out. But in order for you to understand it, I'm going to cut all the jargon. This is the first time I've ever done anything like this. It's an experiment. Everybody from my workplace and home thought that this was impossible, so we're going to try to prove them wrong. It's a really exciting time in biology. It's almost an out-of-body experience to be practicing this right now because things are changing so quickly. There are no themes that cut across what has happened over the last few years other than one, which is that some of the most impactful and meaningful things have come from completely wild and unexpected places. So this is a story like that, and it's very early, and now you'll have heard it, and we'll see where it goes. So the question that we started out with is shown at the top of the slide. It's a basic science question. We had no applications in mind. It was a puzzle. If I asked you, after having been through the pandemic, what is your immune system thinking when it sees a pathogen like a virus? We might argue a little bit, but we would eventually agree largely that it's trying to stop the replication and spread of the virus so you don't die. That's why your immune system exists. So then, let me ask a different question, which is, what is your immune system thinking when it sees the friends, the microbial friends that live in your stomach and on your skin and in your mouth? And that question has been puzzling, and I'm going to try to convince you that the answer is bizarre and may end up being useful. The immune system, you might think, would see our microbial friends, which coat all of the exterior and interior surfaces of your body, and give a friendly wave and walk in the other direction because they're there to help us, and so we shouldn't attack them. But that's not at all what happens. What happens is completely crazy. I'm going to show you what happens. All of this work took place before we got involved. What happens is that certain microbes, when you put them on the skin or in the gut of a mouse or a human or a monkey, induce a huge burst of immune cells on the other side of the barrier. So a few things that we've learned about this process. The first is that the kind of immune cell that comes out the other end of this sort of bizarre process seems to be dictated by the bug who's on the outside. We have no idea how that works. We still have no idea how that works. I thought that was the problem we were studying, but it turns out we're studying something completely different. The second observation is that the immune cells that come out the other side of a process like this know exactly what they're looking for. They have a receptor on their surface that is completely specific for a piece of the bug who's on the other side of the skin or on the other side of the epithelial lining of the gut. So they're entirely specific. You wouldn't be surprised to see this from an immune cell that's responding to a viral pathogen, but seeing this in the context of a microbial friend is a little alarming because it looks like the immune cell is on the hunt against a friend. And then that brings me to this third odd thing, which is that ordinarily when you get a big adaptive immune response, something has broken through your skin. Either you got vaccinated or a pathogen got in, and then there's a 5-alarm fire, and these early responding immune cells rush in, there is swelling, you feel like shit, and that is sort of what your immune system is supposed to do, and hopefully that early response summons a big second response by your immune system, which is entirely specific and clears the pathogen. That's what's supposed to happen. What's weird about this is that when your immune system responds to one of your microbial friends, apparently nothing happens. You can't see any signs of inflammation, there's no swelling, there's no nothing. It's just that about 10 days later, a big burst of adaptive immune cells show up. And then, puzzlingly, they do nothing. They just sit there right underneath the skin or right underneath the epithelial lining of the gut. So why are you getting this huge immune response that does nothing across seemingly an impenetrable barrier? What happened? So that's where we got involved, and I'm going to tell you about a crazy experiment that a physician scientist in my lab did. And I'm going to tell you what she did, and then I'll explain why she did it. It works better in that order. What she did was, this is a massive screen by my academic standards. What she did was to genetically engineer a skin bacterium, a bacterium that lives on every square centimeter of skin of every human on the planet. So this is a friendly bacterium, and she put a little piece of a tumor inside of its genome, so it would make it on the surface of the bacterial cell. Why did she do that? Because she wanted to trick the immune system into making immune cells that the immune system thinks are specific for the bacterium, but we know are also going to have the same specificity for a tumor that resides elsewhere in the mouse. Why did she want to do that? Because she wanted to put these immune cells to the test. She didn't believe that they're there to do nothing. She wanted to know whether there's only two possibilities. One is that they would sit and loiter right underneath the skin where she had put this engineered bacterium and do nothing, which is all anybody had ever seen them do. The other possibility is that they would do what any self-respecting immune cell should do, which is to leave the site of microbial colonization, travel to the tumor, and kill a tumor cell. You understand the distinction? So then, I'm going to tell you how this experiment works because it's unusual. It's very simple. Most of the experiments we do are really complicated. This shows you what we do when we genetically engineer things. This physician scientist in my lab, Erin, did not think any of this was going to work, and so she made dozens of different versions of this engineered bacterium, thinking that none of them are going to work, so she might as well try everything. And so then she did this experiment. The experiment, the technical details are shown here, but let me just tell you that she used mice that are not engineered in any way. These are normal lab mice. They're genetically completely normal. And they have a tumor that's sitting in their flank, their hip. And all she does is to take a Q-tip and dip it in a culture of this engineered bacterium and then very gently rub it on the head of the mouse. That's it. That's the entire experiment. Just a gentle application of bacteria to the skin on the head of the mice. And so she did this, I think, expecting nothing to happen. What she found surprised all of us. If you apply nothing to the skin of the mice, or you take a bacterium that hasn't been genetically engineered, all of the tumors will grow out of control and all of the mice will die. It's a very aggressive tumor. However, if she applies this engineered bacterium that has a little piece of the tumor on its surface, then about 10 days later, the tumor completely goes away. So this is a shocking result. Like all good data these days, it arrives as a text message, usually in the middle of the night, and there it was, a whole line of mice that had no tumors in them at all. Okay, so we eventually figured out how this worked, and what's happening, and how this puzzle of sort of seeming action at a distance is that the bacterium that sits on the skin is inducing a very powerful immune response. We hadn't realized that it was as aggressive as it was, and that immune response mostly takes place in the lymph nodes that drain the skin on the head of the mouse that sit right around here. And then those immune cells, having been trained, rush through the blood vessels in the mouse's body and go right to the tumor and kill tumor cells. We had no idea that they were capable of doing this. In fact, this works well enough that the most aggressive form of tumor we could put into these mice, we let it grow for as long as you can let this tumor grow for, which is about 2 weeks. They form these large, palpable tumors that you can touch with your finger and are probably about a week from killing the mouse. And then we apply the bacterium. You can see on the next slide that the tumors, each one of them will grow, and then when the bacteria show up in the skin, they immediately shrink. So this is something that I think the surprise here, and the reason why this work got so much attention last year, was because nobody had expected this degree of aggression from the immune response to a friend. So I think we had it all wrong. We thought the immune system had friendly things in mind when it saw our microbial colonists, but it doesn't have that at all. Okay, so then I'm going to tell you one more mini story, because where this work has gone, I think, is even more mind-boggling. A different trainee in my lab, whose name is Jeanette, she noticed that when the immune system responds to a pathogen, typically uses two different arms of the immune system to do that. The one I just told you about, which involves killer cells, is shown on the right. This is called the T cell. You don't have to remember that. The one on the left is usually invoked at the same time, and that's something all of you know well because we talked about it all the time during the pandemic, which is antibodies, which are made by a different kind of immune cell called the B cell. So Jeanette wondered, is there anything going on with antibodies here? I can't believe we had all missed this, but Jeanette did exactly the same experiment that Erin had done. She took a Q-tip and dipped it in a bacterial culture of this skin bacterium and put it on the head of the mouse and walked away. I have to tell you something that I think you remember from the pandemic, which is that when you get your first shot, there's a big burst of antibodies that show up in your bloodstream, and then they fall down. And then you come back 3 or 4 weeks later for your second shot, and then there's another big burst, and then it kind of falls back down to a slightly higher level. That's the way vaccination works. That's what we're all used to seeing. When she put this bacterium on the skin of the mice, what happens is rather extraordinary. The level of antibody doesn't go up and down like that. It just goes up and up and up until about 6 weeks, and it just levels off, and it stays there forever. So it's as though the mouse has now been vaccinated against this bacterium that was put on its surface. And so we've learned a couple of things about this, and I'll show you how we're trying to make this useful. We could see that the antibodies that were elicited by this process of colonization were targeting something that sits on the surface of the bacterial cell. So Jeanette figured out, using kind of clever molecular techniques, what they're targeting. They're targeting a giant tree-like protein that sits on the surface of the bacterial cell and sticks straight up, straight into the air. And there's a lot of interesting things about this. We're not exactly sure why the bacterium wants to do this, but it very potently induces the immune system. So for some reason, the bacterium is trying to goad your immune system into developing antibodies against it. Again, we have no idea why that is. Jeanette figured, though, that this could be useful. If this thing is so potent at inducing an immune response, she wanted to put a different protein into this position. Why did she want to do that? Because she thought that it might be possible to create an unusual kind of vaccine this way, where whatever you place here, we could ask your immune system to make a lot of antibody against that. So I'm going to show you how she did that. She figured that if you put something that's not usually there on the surface of the bacterium, then by painting it onto the head of the mouse, we could encourage the mouse's immune system to make a lot of antibodies against whatever the orange thing is. And what she used, because we use it all the time in immunology, is a fragment of the tetanus toxin. You guys have all been vaccinated against tetanus, so she used the thing that you were vaccinated with to see whether that would work on the surface of the bacterium. When Jeanette made this engineered form of this skin bacterium where she put the tetanus toxin into this position and then painted it on the head of the mice, she elicited an extraordinarily high level of antibody against tetanus in the mice that had been colonized. So I have to show you that this is the kind of data point that captured our attention fully. When she challenges the mice with tetanus toxin, all of the mice that have had this bug painted on their head live, and all of the other mice die. And I'm going to show you this because this is the moment where we realized we really had something. We'll see if this video works. This video does not work, but I'm going to tell you what it would have looked like. These mice on the left were challenged with tetanus toxin, and it's a little bit grim, but having been challenged with tetanus toxin, the mice will shiver for about 90 minutes because they're having trouble moving the muscles that control their breathing, and then they will die. The mice on the right are running around very happy, having been treated with the test article, this engineered bacterium with a little tetanus toxin on its skin. And this was a shocking result for us because, remember, all that happened to the mice that were unaffected by being injected with tetanus toxin was that they had a Q-tip rubbed on their head and then Jeanette walked away. So I think what's most exciting to us is the idea that we originally went into this project trying to understand what the immune system was thinking when it sees a bacterium that lives your whole life on your skin. And what she came out with is an approach to vaccination that could be incredibly powerful. The way we apply this bacterium to the skin of the mice is we whip it into a little bit of cream that we buy from CVS, Cetaphil. Some of you may even use Cetaphil. Imagine taking a little tab of Cetaphil and rubbing it behind your ear, and that's it. You're vaccinated. No needle, no healthcare worker, no pain, and for those of you who have children, no need to hold a screaming child down to get vaccinated repeatedly. We think this could be not just incredibly cheap and easy to distribute, but a very powerful way of eliciting a kind of immune response that doesn't just show up in your bloodstream, but also in the membrane inside of your nose, in the membrane on the inside of your lungs, which is where you really need immunity against respiratory pathogens like SARS-2 and flu. And so, that's the rest of my story. If anybody has a question, I would be happy to answer it, but it's a tale about how unexpected things can emerge quickly in biology these days.
Have another hand for Professor Michael Fischbach. Great stuff. That was phenomenal. I don't know about you. For everyone who sat through